Good afternoon and welcome to today's International Myeloma Foundation webinar entitled Top Myeloma Research Presented at ASCO and EHA 2024, moderated by Dr. Brian Durie. Dr. Durie.
Well, thank you, Becky, for that introduction. I'm very pleased to welcome our guests today for this Top Myeloma Research presentation. First of all, I'd like to welcome Sherry Baker, a patient advocate who comes from close to Boise in Idaho, Treasure Valley, which sounds actually very wonderful, Sherry. And then all the way from Denmark, we're welcoming Karsten Levine, who lives just outside of Copenhagen and is the chairman of the Danish organization there in Denmark. So welcome to both. And the important part about today's presentation is that we are talking about the top myeloma research with a close focus on the patient perspective. The perspective of these two advocates who are right with us here, and also those of you who are listening who can communicate in the chat box, and we'll be trying to pick up your questions as we go through each topic. So if you have a question about a particular topic, please go ahead and put that straight into the chat box, and we'll try to pick up on it. So these are the top 10 topics that have been selected for today, and I'll be going through these one at a time, just with some few details and an opportunity to discuss, all the way from Iceland to what is the future like and how close are we to curing myeloma. And so the first topic is about Iceland, and many of you who stay alert to what's happening in the myeloma community have probably heard a number of presentations and comments about the iSTOP-MM study. Iceland screens, treats, or prevents multiple myeloma, and this is a large study led by Sigurdur Christensen and his team of about 30 investigators now, based in Reykjavik, Iceland. And the project is based upon the screening of people over the age of 40 to see if they have a myeloma protein present in their blood, what would be called MGUS, smoldering myeloma, and what turned out to be the occasional case of active myeloma that showed up when the screening was conducted. There was a tremendous accrual to this particular study, with over 80,000 individuals in Iceland agreeing to participate in this randomized study, in which there are 3 arms, one in which no further workup is determined if there's a monoclonal protein found, one in which there are some periodic follow-ups, and then another where a lot more testing and follow-up is conducted. And in this first slide here, you can see that when the population over the age of 40 was screened in Iceland, over close to 4,000 individuals with MGUS were detected, with over 1,000 in the intensive arm, with more detailed testing and follow-up. And so in the recent meetings, and particularly at ASCO and EHA, discussions focused on 4 of the most recent findings from this very interesting project. One of them, which was presented at EHA, is that in looking at the weight of the individuals who were found to have MGUS, it was noted that there was a significant linkage with being overweight. And in fact, the more overweight the individual was, the more likely they were, in fact, to have MGUS. So a very important correlation there. Studies have also shown that despite what has been seen in patients evaluated at the hospital or in the clinic, using these screened patients, there was not a link to other types of autoimmune disease. Perhaps one of the most helpful findings has been that using some simple test is possible to predict whether a patient with MGUS might have more than 10% plasma cells in the bone marrow. In other words, whether it's necessary to actually do a bone marrow or not. And I think that what can ultimately be perhaps the most significant finding is that when patients are screened, they are followed more closely, and at the point of developing myeloma are much less likely to have developed symptoms and features of myeloma, such as the CRAB criteria, with increased calcium, kidney problems, anemia, or bone involvement. And so my first question, and I'll start with Karsten, what is your reaction to the iStop project and also these most recent findings?
From a patient perspective, I would say that we already know that early diagnosis of active myeloma is extremely important to avoid the CRAB fatalities. And it is now very interesting to find out whether very early diagnosis, even before there's active myeloma, and starting up treatment of some kind in those early lines will actually either be able to postpone the time when active myeloma is present, or perhaps even lead to a possible cure for those who are diagnosed very early. I think that's extremely interesting from a patient perspective. Also, and I understand that screening the entire population of larger countries will be a problem, but isolating which parts of the population that might need screening could also be a very important outcome.
Absolutely. This latter point, I think, will be very important moving forward, looking at patients over a certain age. And it seems like now, perhaps focusing on those who are overweight and maybe some other risk factors. So, Sherry, any immediate reactions from you?
Well, I thought the obesity link was really interesting. It just sort of, I think it emphasizes how important all aspects of our health are, whether we have cancer or not. I know Dr. Urvi Shah has done a lot of studies and a lot of presentations on our health, our food, what we take in. And obviously, if obesity is linked to MGUS like that, that's potentially one way to prevent or delay at least active myeloma.
Absolutely, absolutely. And I do believe that this is one of the ideas, is that obesity is what's called a modifiable risk factor, which means that there can be some interventions to try to reduce that and see what is the impact. Maybe it can lead to reduction in progression of the disease. But anyway, very, very exciting, and lots of diverse findings, and many more to come. In Reykjavik, there is also the Decode Genetics facility, and one thing that we're really waiting for is to see what are the linkages between MGUS, smoldering, and active myeloma, and the underlying genetics of these individuals versus the regular population of Iceland. So much more to come. So if we move forward, a lot of discussion about this ability to intervene early and maybe have an option to cure myeloma. To be able to do that, you need to be able to accurately predict, is the disease going to progress? And so we have been looking at this using a risk score called the 2-20-20 score, based upon the free light ratio of 20, the M component level of 2, and then the bone marrow percentage of 20. Those are cutoff values. Above those values increase the risk. And if you have all of those, and particularly if you might also have some abnormal FISH abnormalities, then you fall into this score of 12, which is the top line there, which is showing a more rapid development of myeloma from high-risk smoldering within 12 to 18 months. But there have been some concerns that maybe this is not accurate enough and a good enough predictor, particularly to intervene with more aggressive therapies. And so there was a desire to come up with a new model, and it was exciting to see Dr. Shaji Kumar from the Mayo Clinic present a new dynamic model this year at the MWG and at our various meetings. The dynamic score for progression is based upon changes in the myeloma protein level, the free light ratio, and the hemoglobin obviously dropping over time, in this case, 6 months. If you have all three of those things happening, the line drops. In this case, the dropping of the line indicates that patients are moving on to develop myeloma. So I do think that this is quite a helpful add-on to the 2020, which fits in with the monitoring which has been happening in the clinic. And so, Sherry, what is your reaction to this? A useful addition, you think?
Oh, definitely, definitely. Especially, looking at it over a 6-month period. I think it's very beneficial.
Yes, yes. And Carson, to your point, that it can allow us to be a little more confident if we're looking to intervene and try to do the very best with our treatment options.
Dr. Carey, we do have a question in the chat
right.
based on the iStop study.
Yes, I saw that. Yeah, about the obesity. I'm not aware of the percentage of obese patients in Iceland overall versus in the iStop project. That's a follow-up question that we need to ask. They only presented so far the increasing weight levels within the iStop population. So I don't have an immediate answer for that. I'm sorry about that.
Thank you.
Okay. So let's move on to our next topic, and a lot of discussion at both ASCO in Chicago and the EHA meeting in Madrid about the best frontline therapy, the best first treatment for myeloma. And the big question was, do 4 drugs, what's called quad therapy, work better than 3 drugs? And really, a lot of information that we've had, not just at these meetings, but previously at ASH and ASCO last year, looking at the combination of 4 drugs versus 3 drugs. And in this case, there were 2 trials presented. Isatuximab, which is an anti-CD38, similar to daratumumab, combined with Velcade-Rev-Dex, and then also compared to VRD as well as isatuximab, Revlimid, and Dex for the older patient population. For the transplant-eligible patients, the IMROS study, which is on the left, there was clear benefit with improvement in the PFS as well as the amount of MRD negative achieved using the 4-drug versus the standard of care VRD. In the benefit study, the 4-drug did improve the MRD versus the 3-drug, but much earlier, and the overall benefit unclear at this point. Now, to put this in perspective, at ASH, what we had and with update presented at EHA was that with DARA VRD, very similar to isatuximab VRD, we also have a benefit of the 4-drug over a 3-drug, which this is the Perseus trial, and with a very striking improvement in the MRD at 3 years, 64% over 31%, 4-drug versus 3 drugs, and with clear remission PFS benefit in that study. So really strong indications that 4 drugs are better than 3 drugs. In the more elderly, in the isatuximab trial, I think we're waiting to see, and in that case, the 3-drug combination is so good, there could be options with the 3-drug versus the 4-drug. But Carson, what is your feeling? Should we be aiming for 4 drugs now?
Of course, it's depending on the side effects of adding the extra drug to the treatment. But I would also say that some of these results show that for the elderly groups, those who can't receive high doses chemo with stem cell transplant, we are seeing very good results for that group now, and that's very interesting. Also, maybe I may be too early, so I'm asking the question back to you, Brian Durie.
Yes.
Could we expect to see that we have the question of whether you should actually take high doses and stem cell transplant versus the best possible for medication combination?
Without the transplant?
I'm just asking.
Yes. No, I think that this is also an important question. I mean, up to now, we have seen, for example, in the IFM study and some other studies, that there is some improvement in the remission or PFS with the addition of transplant, but it's been hard to demonstrate that there's a true overall survival benefit with the addition of autologous stem cell transplant. So this has become a valid question, and as we move forward, there's now the opportunity to see, rather than autologous stem cell transplant, maybe it could be good to think about one of our immune therapies, such as CAR T cells or one of the bispecifics. And so, Sherry, what do you think about 4 drugs? I mean, it's a lot of drugs and more expense, more toxicity. You think we should move towards that?
Well, I think the biggest issue could be expense. I have several patients in my support group who have done the DARA VRD with success, and I don't believe they've had trouble getting it approved by their insurance.
Okay.
Toxicity-wise, I mean, personally, I've been on DARA for 4 years, and I have not had any issues with it. So to add that on top of VRD, I don't know that it adds a great deal of toxicity.
Right.
That's just my personal opinion. I think it's nice to have the isatuximab as another anti-CD38 as a different option for either a triplet or a quad. But I remember when the first years I was at ASH and everything was adding DARA to whatever, and it made it much more effective. So I think if you can tolerate the quad, I think it might get us closer to, especially at newly diagnosed, could get us closer to a cure.
Right. And that's definitely a key point. In this Perseus study, which I have still up on the screen, that 64% was actually sustained MRD negativity, which is something that can be sustained for a number of years. And so I do think it's moving us step by step towards long remissions and potentially starting to cure some patients. So from that perspective, quite important. It's great to see these long remissions. Okay, so one other thing that has been a big topic since April is the fact that the use of MRD to assess benefit has been recently approved by the FDA at an ODAC meeting that happened on April the 12th. And just to go quickly through that, the team that worked on that did include myself, plus individuals here, Nikhil from Boston, Jesus San Miguel from Spain, Bruno from Spain, and Dr. Sheath from the Mayo Clinic, plus Dr. Anderson. This team worked on this project with our academic colleagues from the US and Europe, as well as our pharma partners who contributed studies. And so this was really a major project that led to this 12-0 ODAC vote in favor of achieving MRD negative as an early endpoint to indicate benefit. And so basically, it was based upon individual patient level data and individual patient level data from over 12,000 patients, and the vote was taken upon what's called a composite endpoint. So patients had to have a traditional CR and be MRD negative at least 10 to the -5 at 9 or 12 months to be reasonably likely to predict clinical benefit. And so what came out of this was a cutoff, which was 10 to the -5 threshold, picking 9 or 12 months as a time point, which is good to evaluate MRD, and then the ability to test this in 3 different settings: newly diagnosed transplant eligible, newly diagnosed transplant ineligible, as well as relapse refractory myeloma. And each one of those, you can see that the blue line at the top is indicating superior sustained remission versus patients who are below the 10 to the -5 threshold. And so this is a really important point where this can be used in clinical trials and really raises the bar for new drugs moving forward to see if there is an increased amount of MRD negativity which is achieved. And so I don't know how this strikes either one of you. Maybe Carson, again, you'd like to go first on this one?
Indeed. From a patient perspective, of course.
Yes.
I think this is one of the most important new developments. And at the end, with an approval from FDA and later from EMA in Europe, this could mean much faster access to new treatments. Because at the moment, some of the authorities that locally in the individual countries have to approve the treatments are actually trying to say we need to have overall survival data. And when they do that, we have a very sad situation, because while myeloma patients tend to live much longer, I would say perhaps 5, perhaps 7, perhaps up to 10 years on average, then it will take a long time to get the overall survival data needed, which would actually mean that new treatments would be waiting for 5 or 10 years before we have the data needed. So this is a very important step forward.
Thank you for that. Yeah, this is one of the key points. By accepting an endpoint at 9 or 12 months, this is huge, because with these fantastic responses, remissions can be 5 or 10 years. And so it speeds up the drug development process enormously, which is tremendous for patients, is tremendous for everyone, and reduces the expense and the early access to drugs. And so, Sherry, I would assume that you agree. How do you feel about this?
Yeah, oh, I totally agree. I applaud all the work that went into this to get this approved, and it is nothing but a huge accomplishment to help us patients.
Yes, thank you for that. Yeah, it is. Now, to move forward here, what I want to emphasize is that this is within clinical trials. What we're still struggling to learn and have enough data about is the regular use of MRD testing in the clinic. And it was good at ASCO to see a trial where they looked at MRD negativity in patients on maintenance. And in this trial from the University of Chicago, they stopped the maintenance based upon sustained MRD negativity, and the patients, 40 MRD negative patients, and in this case they used a 10 to the -7. The 3-year PFS was 85%. So using this sustained MRD negativity, they were focused on 10 to the -7, excuse me. This meant that it was pretty reliable to say that patients could do well and stay in remission. And so this is just the first of what we need of many studies to look at something very, very important is if we have 2 years of maintenance, let's say, and we have sustained negativity, can we safely stop the maintenance and have time off therapy and have that benefit? And so, Sherry, do you want to comment on this first? This is the first of what I think will be a number of trials.
Well, I think that's probably the biggest question that patients ask is how long do I have to be on maintenance? When can I stop maintenance? So
Right.
if we can use this information to safely say, this would be a good time, you can take a drug break now, or you can stop until
Right.
you maybe progress again. I think it's huge for patients.
Right, right. And so this is a small study, 47 out of 83. However, I think that people were making note, as you say, for something that is going to be quite important. And so we're waiting for more and more studies like this in the clinic to show with MRD negative, can you safely stop? And then conversely, we're looking at more and more data about if you are persistent with an MRD positive result, what are the options? Do you need to change therapy and the like? And so in the clinic, we do have a ways to go in terms of decision making. So let me move ahead here.
Dr. Durie, there was one quick question in the chat.
Yes.
Number 12. So what would be the expected reduction time for clinical trials due to this new approval?
Good question. Years. So instead of waiting, as Karsten said, a minimum of 5 to 10 years now, these remissions with a quad therapy, or you can imagine a quad therapy plus intervention of a CAR T or a bispecific, a patient could be in remission for in the 5 to 10 year range. And now, if they have MRD negative at 9 to 12 months, we could have a decision that would indicate that longer benefit. And so really many years of potential benefit. You're right, good question. Okay, so let's go ahead. A lot of discussion about DREAMing, the DREAM-7 trial and then the DREAM-8 trial. These are trials which are focused on the use of belantamab mafodotin. And so this is a combination of an anti-BCMA antibody combined with a drug, mafodotin. So it's what's called a drug conjugate, which was actually on the market in the US. However, a longer-term analysis using it alone failed to meet its statistical endpoint. And so these new studies, DREAM-7 and then DREAM-8, I'll show you in a second, looked at combinations combined in this case with bortezomib and dexamethasone versus daratumumab. And so really quite a strict test comparing belantamab versus daratumumab. And then in the other trial, trying to get this to move forward here. Oh, there we go. There we go, DREAM-8, yes. The belantamab was combined with pomalidomide and dex, really actually quite a good combination with pomalidomide obviously by mouth versus pomalidomide versus Velcade and dex, and again with benefit. And so the big discussion at these meetings was Bela is back, that this does show that there is value in these combinations, Bela plus Velcade and dex, and particularly Bela plus pomalidomide and dex. And so topics of discussion were that we are now much better equipped to manage the side effects, which are the eye side effects, what's called keratopathy, which has been a concern for patients, this impact on the ability to see clearly, which has been quite a worry. And two aspects to that. One is that we have better ways to monitor that easily, plus by spacing out the dosing of the Bela it means that we can space out the risk of this keratopathy with spacing out to as long as 12 weeks, for example. And so there's enthusiasm that although it will take a little bit of time to get regulatory approval, this is going to be a new option back again. And so Karsten, what's your perspective? I do believe that Bela did continue to be available for a while in Europe as well.
It was only available here in trials, not a standard treatment, to my knowledge.
Okay.
And I have heard about patients who had good results, especially when no other treatment was available. So it is probably a very positive outcome that it is becoming available again with better handling of the side effects.
Right. And Sherry, what's your reaction? It's an anti-BCMA therapy, which is much more easily managed than CAR-T or some of the bispecifics, for example.
Well, I agree with what Karsten said. Some people did have good success with it. And so if it can be adjusted for the toxicities and still be effective, it's good to have it back. And especially if it's doing well in combination now.
Right, exactly. Yeah, so we're optimistic about that, and it'll take some time to go back through regulatory approval, and so we're kind of waiting to see the details of that. But there was quite a lot of excitement about the availability of belantamab. Okay, so a lot of excitement about CAR T cells, and I'll be interested in the reactions to this, but we can comment more broadly about CAR T cells. One of the concerns has been the access to and the availability of CAR T cells. We have 2 commercially available products available right now. What was presented at the EHA meeting was a new type fast CAR, which is one that was developed in China and was recently taken over, bought by a large pharmaceutical company, AstraZeneca. And Dr. Wan Du, an investigator at the University Hospital in Shanghai, had the opportunity to study this fast CAR product as a first-line therapy in patients with newly diagnosed high-risk multiple myeloma. And so those are patients that received VRD as an initial induction, usually 2 cycles, but could be as many as 4. But mostly, I think all but one received just 2 cycles of VRD. And then this product, which can be manufactured much more quickly and can be available for administration very fast. And since it's being harvested much earlier, these are much better T cells which are in much fitter and have what's called a young phenotype and much more likely to be active against the myeloma. And very encouraging early results. Well, you can't do better than this. 100% MRD negative at 10 to the -6, and well tolerated with low-level cytokine release syndrome. So we await follow-up results, but quite encouraging to see this type of a new innovation. So, Karsten, what's your reaction to this type of a CAR T product and CAR T overall?
First of all, myeloma still being an incurable disease.
Yes.
Any new development with especially CAR T and other immune technologies are very important, because I've heard from a lot of your colleagues, Dr. Brian Durie, that they expect and hope that this could be the way to a cure. Also, if I may just very shortly add another issue.
Yes.
Already now, CAR-T is showing very, very good results. Cilta-cel is showing very good results in Germany. Also, a lot of Danish patients are actually paying to go to Germany to get it.
Right.
I have an interesting question. They're coming back here, not going into hospital. Is it actually so expensive when you compare to the fact that you may get years without being at hospital and without continued treatment? Sorry for adding this question.
Well, no, this is a very, very crucial point. I think that the logistics of CAR-T and the expense, key point. So you have this upfront expense, but a high level of dramatic benefit. And then when the patient comes home, they're in remission. And for now, at least, many patients are going without any therapy on a treatment holiday. And so this is really fantastic that, yes, you have that upfront expense, but you do have this potential of dramatic benefit and time off treatment for potentially a very long time. And so I think that my feeling is that we really do need to more fully explore this potential and make it more widely available in the appropriate settings. So Sherry, what's your experience with CAR-Ts so far in your group?
Surprisingly, it has become more accessible. There was a while there where they just couldn't keep up with the manufacturing and there weren't very many slots available. But, I'm in a small area, relatively speaking, as far as population and a small support group, but I've already had at least 3 patients in my support group who have had CAR-T. So, but those have been after, in a couple of cases, many treatments. So
exactly.
The 6-week waiting time for the manufacturing was difficult with the bridging therapy. So if you can have this fast CAR where they're produced quicker, and now that CAR-T has moved up to earlier lines, maybe like you say with the T-cell exhaustion, having
Right.
it newly diagnosed, if it can become available, but at least at second line, I think that gives us hope for better outcomes in the long term of overall survival with CAR-T. But I don't know how big this study was with the fast CAR-T, but you can't beat those kind of results. And to produce it quicker, there's just so much to be said that's positive about this.
Right. This was a small study, and we're just waiting for the longer. There's a question in the chat box as well about the longer term follow-up, but this study is out with the first patients in the 3 to 4 year range already. And so it does seem that the benefit is sustained at least in these early years, 1, 2, 3 years. So I'm optimistic that this in the early use can be of sustained benefit, but just one of the kind of exciting areas. And as most of us have heard, is that there are a number of trials going on and planned looking at this product, but also a lot of other products in addition to the ones that are already commercially approved in the earlier disease setting to see how much benefit that we can garner with that. But we're learning how to reduce the side effects, and it's really remarkably promising. Okay, so I'm just seeing if there's anything else.
Dr. Durie, number 22, please.
Number 22, yeah. Yeah, so there are studies ongoing to compare CAR-T with autologous stem cell transplant, and so those are going to be particularly exciting. There's I think it's KarMMa-9 study that's going to be going forward or is going forward. So there are definitely trials where we'll be able to see, and I know that a lot of people are keen to see the last of autologous stem cell transplant, that high-dose melphalan, which we know does have benefits, but also a lot of side effects. And so people are quite keen to see if maybe we can move away from something that has been a mainstay of therapy for 30 years, but really with a lot of impact on the patient level. Okay. So another abstract which got some attention is this one about teclistamab, which is obviously a bispecific anti-BCMA monoclonal antibody. So it's a bispecific with one arm attaching to BCMA and the other to the T cells to have that combined immune effect against the myeloma. And teclistamab has received both FDA and EMA approval. And so we do have real-world data. And the question came up, if we try to reduce the toxicity in the real-world setting, does that impact the response and the benefit? And so there was a lot of interest in this particular abstract, which looked at reducing the schedule from weekly to every 2 weeks or once a month to see if the benefit would be retained, and it was, with results similar to the baseline Majestic trial. And obviously, the risks of toxicity, particularly infection accumulating over time, was less. And so a lot of interest in this study as an indicator of the direction that we need to be headed with teclistamab and the other bispecific monoclonal antibodies, which are obviously off the shelf, very promising and very active, but with a number of issues, particularly infections and other kinds of side effects and the like. So, Sherry, what's your thinking about this one?
Well, my most favorite part of this abstract
Yes.
is the part that says real world data.
Okay.
Which I think was kind of a lot of abstracts that were presented at ASH this year were real world data. And I think that's so important because a clinical trial obviously is a set group of patients, and you can keep following that specific set group of patients, but there's a lot of patients that are not eligible for that transplant who will eventually be on these drugs one day. So to see, you know, like for instance, if you have a lower renal function, which I have, it precludes me from most clinical trials.
Yeah, right.
So how are we going to know how well teclistamab does for someone, you know, with lowered renal function? So
Exactly.
to start to show the real world data of now we're giving this to the general population and how are they doing, that I think is so important to present. And so I'm really excited to see the real world data coming in on many, many different clinical trials.
Yes, yeah, thank you for that. And so actually this data set is part of a larger project that the IMF is actually conducting with Dr. Tom Martin as the lead. We've started an immune therapy registry to gather up the information as these CAR T and bispecifics and the like are used in the real world setting to see what are the results, what is the best sequencing, is it good if you take one before the other? If you take teclistamab, does that make it bad to try CAR T later, or what is the best real world approach when you have access to these different products?
I think,
yes.
I was going to say, I think that's probably one of the most important questions that patients have, and doctors too, is sequencing.
Right.
Which comes first, which is better to go first? And that's an important question.
Yeah, and we're just struggling with that. But we are looking to get these answers. So Karsten, your thoughts on this?
My thoughts on this is, of course, that the sequencing is important, and also in relation to this, the targets. So the possibility, if you have a CAR T first going after BCMA, then perhaps real-world data telling us whether using a bispecific antibody after that, going for another target, would that be the right way to go? But of course, it will be a long time before we have data. I understand that.
Yes, but we're really working hard to gather the data as quickly as possible. We have a target of getting, for example, data on 6,000 patients, where we're gathering them up from multiple sites around the world to try to rapidly bring that data together to see if we get signals of something being especially good, or particularly if something is to be avoided and not working out quite as well. And I noticed that in the chat box at number 34, number 32, there was another abstract that was presented about we maybe will move away from stem cells and stem cell transplant, but the stem cells that have been collected, there was an abstract at EHA showing that those stem cells can be given to produce quite a nice boost in the blood counts after immune therapy, for example, CAR T or the like, to boost the hemoglobin, the platelets, and to reduce the need for white cells and antibiotics and IVIG. So a boost using stem cells is definitely something to be considered. Okay. Now, another area where we haven't heard a lot about these agents, but these are agents which are called the cell mods, and these are the two which are under development now by the BMS company, and they have what's called enhanced cereblon binding versus the IMiDs, which is versus Revlimid, pomalidomide, and the other IMiDs. And so the diagram shows that this cereblon binding binds over to give potentially a stronger effect on what's called this sensor loop. And so there is hope that these cell mods can definitely significantly add to what we're currently achieving. Next slide. And so there were a number of abstracts presented at ASCO and EHA looking at different combinations, and my reaction is that it's still a little bit too soon to know. Obviously, we are seeing activity of these cell mods, which are similar to the IMiDs, in patients who have become refractory to Revlimid and pomalidomide. However, for me at least, it's still a little bit unclear what will be the major niche or role of these new cell mods, although they're exciting and interesting. Karsten, any reaction to these agents?
I have to admit that this is a little bit over my level here, so I'm listening.
Okay. No worries, no worries. Yeah, I think that we just haven't heard that much about them, and I think we're all keen to learn more about what would be the role of these new immune agents. Sherry, any buzz in your group about the option for these agents?
No, not yet. And I'm with Karsten. I know that I have heard of iberdomide and the other cell mods, but it's kind of above my level right now, and it's going to take a lot of studying to understand these.
Right, exactly. Yeah, I think that's true for everyone. And I think that actually, I mean, if you look off to the right there, we're looking at 50% response rates, and I have to say that maybe we're getting a little bit spoiled, where we're looking at these very, very high response rates. I just showed you 100% response rate with fast CAR, and we get 70% easily with the bispecifics and all of these newer immune agents. And so really we are kind of targeting these other exciting agents, I would have to say. Okay, so where are we in terms of achievements, and what does the future look like, particularly thinking about the possibilities of cure? And I've listed a few items here that I think that we need to be strongly considering looking to the future. As was already commented, I think that the access is a priority. We already have 4 drugs in the front line as an option. Earlier CAR T we need to think about. Establishing a niche possibly for some of these other agents, intensification for patients with high-risk disease, and then if we can stop therapy based on MRD, maybe we can limit treatment to 2 or 3 years. Carsten, if you were to prioritize any one of those, what do you think we should be pushing for first?
Coming from a part of Europe where access is relatively good, I feel that I have to say improved drug access worldwide towards populations where even a 2 or 3 drug combination that is standard here and in the US is not available should be a very high priority.
Absolutely. Well, I have to say I'm really in agreement with that. I mean, I as you might imagine, give lectures in different places, and a number of them a great excitement about 4 drugs and CAR T cells and the like, but only if they had access. And so that access we need to work on and make sure that we're paying attention to myeloma patients in the US.
I may add,
yes.
Add in here that, of course, here in Denmark and in Scandinavia, it is access to CAR T together with the bispecific antibodies, which is the number
key
1
thing.
priority. But the international perspective is so important.
Right. I would agree with that. That for example, in Asia, except for China, basically no access to CAR T. Now, Sherry, you already mentioned that although there was a backlog here recently, there has been more ready access to CAR T in the US for both products. What's your thinking on that now?
Well, I think the sequencing of which one you would do first, I think it's terrific that CAR T is now available in second relapse, or I think it's second now. And but when I was first diagnosed, the choices were pretty much Velcade and Revlimid. And so, you know, when I was on one, and we weren't doing I didn't do the combo at the time because of my kidney issues, but I always thought, okay, here's step A, here's step B. When those don't work, you know, what's the third new thing to come out? Well, now it's not that simple because there's so many more options. So I know next time I relapse, I keep looking ahead of what would I choose. But by the time that happens, there's 3 or 4 more options out there. So those waters get very muddy. And so I think that's the biggest, for me, the biggest thought of the future is what, you know, the cart before the horse, and what comes first and what can you do later? What should you save for later?
Right. Yeah, these have always been questions. I think we've been most fortunate that actually there have been a lot of amazing new therapies, especially these new immune therapies, like coming later. And so there is not that same need to be kind of saving for later. And so now we're looking to bring these newer active agents in as quickly as possible.
Well, that's what Dr. Joe always says: is don't save the big guns for later. You know,
bring them in, and get that big advantage, because these early remissions are the best remissions and can give you that best quality of life. Absolutely.
But one more thing, I do agree with Carsten that access especially in countries outside of the United States, is a huge, huge problem. So I know that needs to be addressed.
Definitely, definitely. And we have some planning on that to see how we can work on that, whether it be with generic agents or with, I think, as actually Karsten, I think, has been involved with working out deals with individual governments so that there can be a more positive approach to drug access and approval within each country, where you need to work with the budget that the government has, and then also with what deals can be struck with the corresponding pharmaceutical company. But one thought I want to just end with is how close we think we are to curing myeloma, and we've had a couple of trials focused on this. One is the CESAR trial, which was using the combination of KRD plus autologous stem cell transplant with maintenance. And this was the study conducted by Murray V. Mateos in Spain. And we now are out at close to 6 years of follow-up. And really, the treatment of high-risk smoldering, in this case, it was based upon the 2-20-20. So patients who were at pretty high risk of progression within 12 to 18 months get KRD plus transplant plus consolidation and maintenance. And so the end result of that is that at 4 years, 40% of the patients remain with sustained MRD negative. And so this is, you know, an important step towards getting sustained MRD negative remissions. And even those patients who maybe have had biochemical progression, which is, so 60% have had some reoccurrence of the disease. If you look to the left, most of those patients have not progressed. And actually, at 6 years, 94% of the patients, some have some recurrence of the spike, but 94% have not actually progressed. And so then if we look at the ASCENT trial, which was adding daratumumab to KRD, and this was a study led by Shaji Kumar and myself, actually. So we looked at MRD negativity of 84%, and those patients are continuing to do well. And so I think that we are encouraged that there is sustained MRD negativity, and we are making some headway towards sustained MRD negative, but also what we can call some sort of a functional cure, where patients maybe have a low level of residual disease, but have not progressed. And so, but we need to have confidence in offering this kind of therapy to patients with early disease. Yeah, but in my perspective, the future is bright. We have lots of options to try and enhance those results, especially with the new immune therapies. And so I think that for the average patient, the future is bright. For new patients coming in, if we treat them early, I think that the future is particularly bright if we have confidence to intervene early. So any final thoughts, Karsten?
I can only say that whether the end result is a cure or turning the disease into a chronic but treatable disease with very few side effects, that's not so important. And I think that the research that you, Dr. Durie, and all the other fantastic researchers are doing is probably bringing us there within a few years. Yes,
and Sherry?
I agree with Karsten. If you know, I don't like calling it it is still cancer, and I don't like calling it a chronic disease, because people are still dying from this. as they are probably with some chronic diseases as well, but it is still a cancer. But if we can treat it, if we can't cure it, but we can keep it at a low level with
Right.
less toxicity and maybe also less financial toxicity,
Right.
then we're making progress. And I agree, the future is bright.
Yes, yes. And so I want to leave you with one other picture, which is it's bright because all these folks right here on this slide, these were the myeloma experts, the key opinion leaders that were at the IMWG summit in June in Madrid. And what can we say? If all of these minds are focused on this project of trying to help myeloma patients, for sure, the future is bright, okay? Thanks to you both for your very, very insightful and helpful comments and feedback today. And we have touched on a few questions. Question number 40 says, Karsten, that they completely agree with you. Making treatments accessible worldwide is a key goal. And this is from a Canadian. So anyway, thank you guys. I hope that this was valuable for our audience. We covered a lot of topics, but appreciate the involvement. And Becky, if you want to close us out, thank you.
Absolutely. As a reminder, a replay of this webinar will be available on our website early next week, and it will also be emailed directly to all of you who registered for this webinar. Next slide. Please remember to fill out the survey that you'll be receiving as you close this window. It will also be emailed to you as well. We really take your feedback to heart, and we make some changes in programs that you guys have requested. So please respond back to that if you would like. Next slide. And next slide. And thank you to our sponsors. This would not be possible without them. AbbVie, Karyopharm Therapeutics, and Sanofi. Thank you so much.
All right. Thank you much for that, Becky. And thanks again to Sherry and Karsten. Have a good day, everyone.